Everyone. Today's lecture is going to explore about the relationship between DNA, chromatin and chromosomes. And the best way to begin is forming a visual with the help of this pasta and noodles that are shown on your picture. And this is how the whole DNA is compacted into your nucleus. It superfolds like this, like the Maggie coil appears, and once the Maggie is cooked, however the cooked Maggie appears in a bowl, this is how it is present inside the nucleus. So I think with this it's a best way to proceed for the lecture. So you know that the nucleus of every cell has DNA, and this is the structure of nucleus on your screen. This nucleus of human cell is approximately 10 micrometers in diameter, and it takes up about 10% of the cell's volume. So the nucleus is the largest organelle in animal cells. The length of human DNA in every cell is around 2 meters, and if we sum up the length of all the DNA that is present in every cell of our body, then it comes up to about 30 times from Earth to Sun and back, whereas it is around 6,000 times the distance from Earth to Moon and back. So you can imagine the total length of the human DNA that is present in our body. Now, because of this large size of the DNA, the cell is posed with a problem. As you know that the nucleus is just around a few micrometers in size. So how would this 2-meter length DNA fit into the nucleus of every cell? And this is what we are going to address in the forthcoming slides. So, the next slide is talking about a special type of protein known as histone proteins, because these proteins are helping the DNA to fit into the small nucleus, as the DNA is too large and the nucleus in comparison to the length of the DNA is so small. So one wonders how will such a large molecule fit into such a small organelle, and it is with the help of these histone proteins. So see, look here, these are the histone proteins, and they are of four types: H2A, H2B, H3, H4. So these four together form the octamer, and then it is followed by H1. So these histone proteins are positively charged proteins that are found in the nuclei of eukaryotic cells, and they play a key role in DNA regulation, replication, and they help the DNA to fit into the nucleus. Okay, so this large 2-meter DNA that every cell of human being is there, it fits with the into this nucleus of 10 micrometers with the help of these uh, histone proteins. So what is the function of these histone proteins? It helps the DNA to coil, it makes the DNA compact, and it forms chromatin. These histone proteins are made up of some basic amino acids like arginine and lysine. Arginine is a hydrophilic amino acid that loves water, and lysine is the most basic amino acid. So DNA is a highly negatively charged molecule due to the large number of phosphate groups present, and the histones are highly positive charged proteins uh, because of arginine and lysine, and they neutralize the negative charge of the DNA. So now we will look at the higher organization of DNA. Now, what does this higher organization mean? It means how the DNA will be compacted into the nucleus of the cell. So, in higher organisms, DNA is organized inside the nucleus. A segment of double-stranded DNA is wrapped around a complex of histone octamer, which is formed by H2A, H2B, H3, and H4. Two of each of these histones will form an octamer, and this is known as nucleosome, which is of 10 nanometer. A group of nucleosome forms DNA fibrils, and the six fibrils supercoil to form chromatin fiber. So you can see here that this is the DNA of the cell. It is lying like a coiled structure. If you want to understand it better, you can imagine like noodles or Maggie in a bowl. So just like your Maggie which you are going to eat is lying in a bowl, exactly like that the DNA is present inside the nucleus. Now, this supercoiling of 2-meter DNA is done by histone, and these histone octamers form the nucleosome when the DNA, a small part of the DNA, wounds 2.5 times around each histone core, and when the DNA wounds around this histone core, it is known as a nucleosome. A small section is free, then once again the DNA wraps itself around the next octamer. So like this, the his DNA wounds around the histone core. The free part, which is unwound or which is free of any octamer histone, is known as the linker DNA. So this is the linker DNA. Now we are going to talk about organization of DNA. So the first thing that happens is the formation of a nucleosome. This is a double-stranded DNA helix on your screen, which is of 2 nanometers. And after the first type of organization, a nucleosome is formed. Nucleosome formation is the first stage of DNA packaging. At this stage, DNA shows bead on string um, arrangement. This can be seen when the chromosome is subjected to partial and gradual unfolding. In this form, chromatin fiber appears like a long string in which small beads are attached at regular intervals. It is of 11 nanometers. After this, what happens is that the DNA further undergoes compaction in 7 folds. The overall compaction requires several 10,000 folds of chromosome, hence a higher order is formed. And it forms a 10 nanometer fibrils, as you can see here, this or 10 or 11 nanometer fibrils. The disc nucleosome has a diameter of 10 nanometer, and the height is around 5 nanometer. They are arranged in a linear manner, and they are packaged. Packaging increases 7 to 10 times. After this, the 30 nanometer fiber is formed, and this is known as the uh, chromatin fiber that is associated with nucleosomes. Nucleosomes basically coil into chromatin fibers. So what happens here that the 10 nanometer fibril is supercoiled into a circular 30 nanometer fiber with 6 nucleosomes per turn. So there are 6 such nucleosomes per turn. Each turn of the supercoil is relatively flat, and faces of the nucleosome of successive turns are parallel to each other. This 30 nanometer formation of DNA provides compaction by 100 folds. Histone 1 protein plays an important role in stabilization of this structure. So please remember that the histone 1 or the H1 is not a part of the octamer. It lies away from the octamer, and it helps in stabilizing the 30 nanometer structure. On further condensation, the chromatin loops are formed. So chromatin fiber are folded into loops, and it is a 700 nanometer structure. Here on the 700 nanometer structure further undergoes folding, and this appears as duplicated chromosome when the cell is dividing. Here you can see each DNA molecule is packaged into mitotic chromosome around 10,000 fold shorter than its full length of 2 meters. This is a so by this time the DNA reaches from 2 nanometers to 11 nanometers to 30 nanometers to further compaction of 700 nanometers and finally to 1400 nanometers. When it reaches the 1400 nanometer folding, by this time the chromosome has folded 10,000 times shorter. Okay, so this is how the folding of DNA and its organization inside the nucleus occurs. So now you know that the DNA is a highly coiled chromatin fiber. DNA supercoiling allows the DNA to compact so that it can fit within the limited space of the cell, and this folding is regulated by special type of enzymes known as DNA topoisomerase. And the DNA, this supercoiling can be either negative supercoiling or it can be positive supercoiling. By positive supercoiling, we mean that the DNA is highly wound or over wound, and by negative supercoiling, we mean DNA is under wound. That means the coiling is opening up. Okay, so this is how the DNA has two types of supercoilings, negative and positive. Heterochromatin and euchromatin. So let's see what these are now that we have understood that the DNA compacts and fits into the nucleus. You will see that the DNA is compressed 8,000 to 10,000 fold to generate the chromosome. The active site of this chromatin that is present in the nucleus is known as euchromatin, and this euchromatin is undergoing transcription or it's preparing to undergo transcription. That is why it is known as the active site. It is less compact, and uh, on the other hand the inactive site is known as the heterochromatin, and this heterochromatin is highly compact, and it's transcriptionally inactive. It has some barriers in its structure that prevent the transcription, and in general it is densely packed, more compact, and has an intact nucleosome structure, and it is resistant to the enzyme DNase 1. It is deeply stained and localized in the nucleus. As it prepares for transcription, it undergoes changes, and to make it accessible for transcription machinery, it is converted into euchromatin. So this is an interconvertible region. And now you can see this is the DNA on your screen, which is getting compacted into the chromosome, and this chromosome has two regions: euchromatin, which is actively transcribing or it is getting ready for transcription, and the densely packed heterochromatin, which is transcriptionally inactive. Now we will see the relationship between DNA, chromatin and chromosomes. So the DNA, now you know, that contains genetic information for an organism's development, growth and reproduction, whereas chromatin is a complex of DNA and proteins that is found in the nucleus of a cell. Chromatin is stable substance that contains about twice the protein as DNA. And chromosomes are egg-shaped structures made up of two chromatids. They are visible during cell division. Chromosomes are condensed forms of chromatids. So this was the relationship between DNA, chromatin and chromosomes.